Hello knowledge seekers, welcome to another video session of Fuzzy Scientist over the topic Metabolism of Carbohydrates. So let me start today's session with a question for you all. Have you ever heard of the word oxygen debt? So basically what oxygen debt refers to is the amount of oxygen required to recover. So by the word recover, what do I mean here? From which point do we have to recover? So to understand this, let us start our today's topic, which is lactic acidosis. And before understanding what is lactic acidosis, let us recollect yesterday's sessions where we have discussed all the 10 reactions of glycolysis. So if you correctly remember, so the glycolysis reaction has ended with pyruvate, formation of pyruvate, which is a 3-carbon compound. Though the though the glycolysis has begun or initiated with a 6-carbon compound, which is glucose, it has ended with a by the formation of a 3-carbon compound called pyruvate. So see, basically pyruvate is a 3-carbon compound. PEP, phosphoenol pyruvate, undergoes a dephosphorylation reaction, which is an example for substrate level phosphorylation also. That means losing a phosphate molecule, it gives rise to pyruvate. So the first of pyruvate form which it gives rise to is in the so the enzyme which catalyzes this reaction is pyruvate kinase. So we get a pyruvate enol form, so this is highly unstable. So as soon as this pyruvate enol form is generated in the reaction medium, it immediately gets converted to pyruvate keto form, which is most stable form of pyruvate. So this pyruvate, if oxygen is abundantly present, enters into the tricarboxylic acid cycle by entering into a second step in the overall electron transport chain system. So, but in the absence of oxygen, what is the fate of this pyruvate? In the absence of oxygen, so this pyruvate, under the action of lactate dehydrogenase enzyme, gets converted to lactate. So, this lactate. Let us now look at into the significance of the conversion of pyruvate to lactate in our body. So basically, as I told you, in the absence of oxygen or in the or under anaerobic conditions, pyruvate gets converted to lactate with the action of enzyme lactate dehydrogenase. So why is it important? See, if we so basically, like, when does this lactate formation basically occur? Let us put some examples. So when our body is under strenuous exercise, or when we are under shock, or what can be another example? So basically, in under these conditions, so what happens is we are deprived of oxygen, and hence this deprivation of oxygen leads to the conversion of pyruvate to lactate. So what is the significance here? Mm, so as this pyruvate, by taking a hydrogen molecule from NADH, gets converted to lactate. So we have to understand here is that this NADH has come up from the sixth reaction in glycolysis, which is oxidation reaction, if you remember properly there. So NAD plus, by taking the hydrogen molecules from the substrate, which is G3P, which is glyceraldehyde 3 phosphate, undergoes gets converts to NADH. So that NADH is used here as um, the conditions right now are anaerobic conditions, that is oxygen deprived conditions. So if you remember correctly, I've already told you that in the presence of oxygen, or if the conditions are aerobic conditions, this NADH would have entered into electron transport chain, contributing for synthesis of 6 ATP. As the conditions that I'm stating now are anaerobic conditions, that is oxygen less conditions, so this NADH reduces pyruvate to lactate with the catalytic action of enzyme lactate dehydrogenase. So this is how NADH is used here. So what is the significance here? As the gets utilized here, so from NADH the hydrogen gets removed and it reduces pyruvate to lactate, and we have now a freshly formed NAD plus. So now this NAD ion is readily used by the cell for for second conversion of G3P to the next compound, which is G3P gets converted to 1,3-bisphosphoglycerate. So G3P gets converted to 1,3-bisphosphoglycerate. So there this NAD acts. So if you remember, so that's the significance of lactate. So what this actually does is it continues the cycle of glycosis even in the even in the anaerobic conditions. That is, even though our body is deprived of oxygen, the glycolysis is continued. So as a newly formed NAD plus is as NAD plus is newly formed in the reaction where pyruvate gets converted to lactate. So that is how glycolysis is continued. So in erythrocytes, glycolysis results in formation of lactate because erythrocytes do not contain mitochondria, which are the centers for aerobic respiration. So not only in erythrocytes, in the retina, in the skin, in the gut or the alimentary canal, and in the adrenal middle of kidneys. So we observe high frequencies of this and lactate formation, high frequency of lactate formation at the end of glycolysis. So now let us look into the next step, which is lactic acidosis. So what does this lactic acidosis means? The over accumulation of lactate in the tissues is simply referred to as lactic acidosis. So what actually happens is, so technically put, overproduction or less utilization, low utilization of lactate or lactate formed leads to lactic acidosis. I mean, see, the general range of lactate in our serum is 5 to 14 milligrams, uh, 5 to 14 milligrams in deciliter of blood. So this is the normal healthy range. So if the range increases more than this, then that can be referred to as lactic acidosis. So now, so what is now let us look into. So what happens in this lactic acidosis is, basically this lactic acidosis can occur due to oxygen deprived conditions, which we observe when our body is undergoing strenuous exercise, or when we experience kind of shock, or in von Grieg's disease. So what is this von Grieg's disease is? So this is a type 1 glycogen storage disorder. So what happens is, in this disorder is, so our cells will not be able to convert glycogen to glucose, which is very much important for the initiation of glycolysis reaction. So in such conditions we observe lactate formation. Okay, so what are the next steps? So see, in in abnormalities, in severe abnormalities like pulmonary embolism, myocardial infarction, and in such conditions we observe a severe deficit of oxygen in our body. So this severe deficit of oxygen in our body results, that too in the condition of cells are deprived of oxygen here, and they do need ATP. As for them to live, the cells are already consuming the ATP that are already present in them, and they do have a high requirement of ATP, and for that they bind to the path of production of ATP in anaerobic conditions. So which eventually leads to the production of lactate, which is lactic acid. So this lactic acid is fatal. This kind of production of lactic acid is fatal to our body. So here this from this point, for us to recover back and become healthy, a certain amount of oxygen is required, and that amount of oxygen is termed as oxygen debt. So in general, in clinical practice, so plasma levels of lactic acid are measured to understand better, or to measure, or to get a proper measurement of the amount of oxygen required to support the patient for his recovery, and so that we can save the patient from morbidity and mortality. So this is the significance of conversion of pyruvate to lactate, and the importance of lactic acid knowledge, lactic acidosis knowledge in us. So oxygen debt. So we have understood what is oxygen debt, we have understood what is lactic acidosis, and we have also understood like what is the significance of conversion of pyruvate to lactate, and what is its importance under anaerobic conditions, and what are the cells that majorly sticks to this pathway in glycolysis. So thank you for watching my video today. So let us catch tomorrow with another topic over this over the major of over the heading carbohydrate metabolism. Until then, see you, and please do not forget to subscribe to my channel Fuzzy Scientist. See you. Bye bye.